Hi everyone, my name is Chet Velker. I will be presenting a molecular epidemiology study that evaluates the link between small nucleotide polymorphisms in the vascular endothelial growth factor gene and glioma risk and prognosis. Gliomas are brain tumors that originate in glial cells, which are cells that surround and support neurons. Gliomas account for about 33% of all brain tumors. Gliomas are classified into three overall groups based on the type of glial cell affected. Astrocytomas affect astrocytes, which are connective tissue cells in the cerebrum and cerebellum. Ependymomas affect ependymal cells that line the ventricles and spinal cord. And oligodendrogliomas affect supportive tissue cells, most common in the cerebrum. Symptoms depend on tumor size and location and include headaches, seizures, behavioral changes, speech problems, nausea, vomiting, and vision changes. In addition to the three classifications, there are subtypes of gliomas that are graded from 1 to 4 by the World Health Organization, with 1 being the least severe and 4 being the most severe. Treatment depends on the World Health Organization's grade and ranges from surgical removal to total resection and chemoradiation therapy. On this slide is a screenshot of the first page of the article titled "Genetic Variants of Vascular Endothelial Growth Factor Predict Risk and Survival of Gliomas." Gliomas can be caused by both environmental and genetic factors. The only proven environmental cause of gliomas is ionizing radiation. Genetic factors involve genes associated with DNA repair, cell cycle regulation, and immune response. Vascular endothelial growth factor is upregulated in gliomas, which promotes angiogenesis in the tumor. Angiogenesis is the formation of new blood vessels. Vascular endothelial growth factor pathways also stimulate secretion of von Willebrand factor, a protein involved in hemostasis, which further promotes angiogenesis and is linked to cancer risk. Single nucleotide polymorphisms in vascular endothelial growth factor and von Willebrand genes are promising biomarkers for glioma risk and prognosis. Single nucleotide polymorphisms, often referred to as SNPs, involve a single nucleotide difference in the human genome that occurs in over 1% of the population. The objective of this study was to evaluate associations between SNPs in 11 vascular endothelial growth factor genes and 1 von Willebrand factor gene with glioma risk and prognosis. To evaluate associations between small nucleotide polymorphisms and glioma, Lienhardt et al. Used a case control study. In the article Observational Research Methods, Research Design 2, Cohort Cross-Sectional and Case Control Studies, Mann states that in a case control study, patients with an outcome of interest are matched with a control group that does not have the outcome of interest. In this study, the case group consisted of 126 glioma patients over the age of 18 that were treated between 2009 and 2010 at 2 hospitals in Braga, Portugal. The control group consisted of 180 cancer-free patients that donated blood during the same time frame. Controls were matched to the case group by age and gender and selected from the same region as case patients. Table 1 provides the demographic information for the case and control groups, along with the frequency of subtypes among the glioma patients. All participants in the study provided informed consent, and the study was approved by institutional ethics committees at both research sites. The DNA used for genotyping was extracted from leukocytes in the blood samples of case and control group patients. Single nucleotide polymorphism genotyping was done on the Sequenome MassArray iPlex Gold system. CD Genomics states on their website that the MassArray platform combines polymerase chain reaction, primer extension chemistry, and MALDI-TOF mass spectrometry techniques to reliably and accurately characterize genotypes. First, DNA containing the SNP of interest is amplified by PCR. Then, a single extension primer extends only the base at the locus of the SNP. And finally, the extended product is measured using MALDI-TOF, and the SNP is classified according to the molecular weight. Several statistical tests were used in the study to compare the frequency, distribution of age, sex, and genotypes of vascular endothelial growth factor and von Willebrand factor, and the allele distribution among the cases and controls. Lienhardt et al. Used the t-test, chi-squared test, and non-parametric Wilcoxon-Mann-Whitney test. The association between glioma risk and SNPs was analyzed using multivariate logistic regression. The test statistic for this test is the odds ratio. George et al. Defined the odds ratio as a measurement of the association between two variables. An odds ratio of 1 indicates no association between variables. An odds ratio of higher than 1 means the odds of the outcome are greater after exposure, and an odds ratio of less than 1 means the odds of the outcome are lower after exposure. Multivariate Cox regression model was used to estimate the relationship between investigated SNPs and overall survival and progression-free survival. The test statistic for this test is the hazard ratio. The National Institute of Health defines the hazard ratio as an estimate of survival in an exposure group compared to a control. As with the odds ratio, a hazard ratio of 1 means there was no difference between groups. A hazard ratio of greater than 1 indicates decreased survival in the exposure group, while a hazard ratio less than 1 indicates increased survival in the case group. Significance was set at 0.05, and Bonferroni's correction was used for tests comparing multiple variables. Linhares et al. Evaluated the association between vascular endothelial growth factor and von Willebrand factor SNPs in risk in glioma cases, as well as glioblastoma and oligodendroglioma subtypes. The investigators chose these subtypes because those were the most frequent among the case group. Only one result was significant after Bonferroni's correction. Since chromosomes come in pair, SNPs can either be heterozygous, occurring in one chromosome, or homozygous, occurring in both chromosomes. The heterozygous vascular endothelial growth factor SNP RS833061 was statistically significant, associated with increased risk of glioma and glioblastoma. The odds ratio for these results were 164.85 and 155.66 respectively, meaning patients with heterozygous RS833061 were more than 150 times as likely to develop glioma or glioblastoma. Other results were significant, including the heterozygous von Willebrand SNP RS9295740 before Bonferroni's correction was used to adjust the p-value. Several vascular endothelial growth factor SNPs were significantly associated with overall survival and progression-free survival in glioma and glioblastoma patients. Overall survival compares the number of patients who have died in the case group, whereas progression-free survival compares the number of patients whose condition has worsened. Homozygous vascular endothelial growth factor SNP RS1005230 was associated with decreased overall survival in glioma patients and decreased overall survival and progression-free survival in glioblastoma patients. Heterozygous vascular endothelial growth factor SNP RS3024994 was associated with decreased progression-free survival in glioma and glioblastoma patients. Heterozygous vascular endothelial growth factor SNP RS2010963 was associated with decreased overall survival in glioblastoma patients. Linhares et al. Demonstrated an association between several vascular endothelial growth factor SNPs and glioma risk and prognosis. Patients with RS833061 SNPs were over 150 times more likely to develop glioma or glioblastoma. Patients with RS3024994 and RS1005230 SNPs had decreased overall and/or progression-free survival. Interestingly, patients with the RS833061 SNP treated with bevacizumab show improved survival. The mechanism for why is still unknown. However, this trend suggests that vascular endothelial growth factor SNPs are promising biomarkers for determining course of treatment in glioma patients. 99.9% of the human genome is identical from one individual to another. Of the 0.1% difference, SNPs are the most common variation. This makes SNPs promising candidates for tracking disease risk and prognosis, or possibly targets for treatment or prevention. The study performed by Linhares et al. Demonstrated that this potential by identifying SNPs in the vascular endothelial growth factor gene that are linked to glioma risk and prognosis. Advancement in molecular technology and techniques have created new opportunities to discover associations between genetic biomarkers and disease utilizing epidemiology studies. Findings from these studies will lead to earlier diagnoses, improved disease monitoring, and novel treatments. This concludes my presentation. Thank you for listening, and I look forward to your feedback.